TB's been around for millennia and been killing people for millennia. The saddest thing about it is that it's both a treatable and a preventable disease. In 2021, over 10 million people fell ill with tuberculosis across the globe, and of those, 1.6 million people died. That's the second biggest infectious diseases killer globally, behind COVID-19. There's a type of TB called drug-resistant TB, and that's tuberculosis that's resistant to the drugs that we normally use to treat it, and that is on the rise. Drug-resistant TB leads to higher mortality, so more patients die, and more transmission of the disease. It's estimated that about 10% of new TB cases are now drug-resistant, and unfortunately only about a third of those cases are diagnosed annually. So a real urgent need for better diagnostics for drug-resistant TB. So back in 2019, while I was still a professor at Quadram Institute, I started developing a targeted sequencing-based drug-resistant TB test in collaboration with Oxford Nanopore Technologies. In 2021, I actually left Quadram and joined Oxford Nanopore. That was to develop this test further within the company. The WHO announced 4 years later that tests such as ours are suitable for the accurate diagnosis of TB, something that would take months using traditional technologies. And this means is that we can use our test when it gets regulatory approval, diagnose patients so that they can receive the appropriate treatment. And this will ultimately save lives and reduce the transmission of TB. So what can happen is that the standard therapy isn't working because you've got drug-resistant TB. And therefore, if you figure that out using a rapid diagnostic that in our case takes only 5 hours and tests for resistance to 16 different anti-tuberculosis drugs, then you can give the patient an alternative therapy so that it works and that they get better.
TB is, of course, one of the great scourges of the world, and to rapid insights in the way that we can diagnose drug-resistant TB is, of course, fantastic news. And to have it endorsed by WHO is really tremendously important.
So our test is designed to work from sputum from patients suspected of having TB. And so this sputum is taken and then we extract the DNA and then we amplify regions of the tuberculosis genome we're interested in, and then when we detect mutations across those regions and we determine drug resistance based on that information. We did a trial with the WHO and FIND to determine the performance of our test. And we did that trial in Mumbai in India, Tbilisi in Georgia, and Johannesburg in South Africa. And we tested about 750 samples, and the results were very good, and that's what the WHO used to endorse our technology. So there are a list of high burden drug-resistant TB settings across the world. China, India, Indonesia, they will be the first countries and the most appropriate countries to use the test in to begin with, but it can be used anywhere really across the world.
It is really important that sequencing be used, I think, in all sorts of diagnostic areas. And it's great to see this being used in the microbial context. And fantastic news, of course, that this is being generated as research done at Quadram. I think we're just at the beginning of a new revolution in the way that DNA sequencing can impact for the better human health.